Today's Grand Rounds is sponsored by the Athena Fund, and our speaker is Professor Stephen Cole from UCLA. Stephen received a bachelor's degree from UC Santa Barbara, and we're happy to welcome back to his alma mater for his PhD, Stanford. The role of stress in health and wellness is often discussed here in Grand Rounds. Steve's research uniquely probes the biology of stress. He pioneered the field of human social genetics, integrating genome-wide RNA profiling into social, behavioral, and population health sciences. Cole discovered the conserved transcriptional response to adversity that describes how social disparities in health serve as a signal for neurally activated transcription of immune cells that result in chronic disease. Cole's work maps the effects of social disadvantage, rising from poverty, discrimination, and social isolation onto cellular, neural, and regulatory processes. Steve's work has resulted in more than 800 publications. His list of honors and awards is so lengthy, it would take up most of the Grand Rounds to go through them. But Steve, it's really an incredible honor to welcome you back to Stanford. Thank you very much for joining us today.
Sure. Thank you. So I, as mentioned, I basically study the everyday life of the human genome, or how the world around us, our social, psychological, political, economic context, basically how everything outside the body that we experience as life somehow gets into the body and it ends up regulating the activity of the human genome. And this is kind of a weird thing to do with our genomics toolkit. So I usually start out my talks by describing how we ended up doing this kind of thing in the first place. So what really inspired this effort to understand those connections were data like these. So these are data from a molecular epidemiologic study in which we were looking at one of the most toxic epidemiologic risk factors for all-cause human mortality and chronic diseases in particular. And basically all this heat plot shows you is that for the set of people who were exposed to high levels of this risk factor, there's probably about 70 or 80 genes that are systematically more active in the white blood cells of these individuals, about 130 genes systematically less active in terms of gene transcription, the rate at which RNA is transcribed off of the DNA genome and ends up structuring the protein complement of these cells. So, it turns out that also these are not a kind of random smattering of 200 or so randomly sampled genes from the 20,000 gene human genome. It actually turns out these blocks of up and down regulated genes represent, or essentially reflect a kind of a, you can think of them as a couple of organized conspiracies or themes. So for instance, prominent among the genes upregulated in people exposed to high levels of the risk factor are genes involved in inflammation, which you guys are all familiar with. So I'm not gonna need to unpack it for you. Prominent among the genes downregulated in individuals exposed to high levels of the risk factor were genes involved in essentially defending the body against intracellular pathogens, things like viruses, so like type 1 interferons, immunoglobulin G. So these kinds of coordinated shifts in you can think of them as the white blood cells sort of molecular portfolio, I don't think would be particularly surprising if the kind of risk factor that we were looking at here were the kind of microbial or physical chemical exposures that we often worry about most in epidemiology. But what was interesting to me about this research is that the risk factor that was structuring these differences in gene expression is just simply the extent to which the people in the first few rows of the heat plot felt disconnected from the rest of humanity. It was loneliness that was structuring these differences in gene expression. And that was a little bit more of a puzzle. We're used to thinking about white blood cells, concerned with microbes. They've got microbe detectors. They're supposed to respond to microbes. They're supposed to defend us against microbes and help reconstruct our bodies when microbes or wounding injuries hit. So why would these immune cells be systematically changing their RNA profiles as a function of something so distant and in some ways abstract as the social experience of the body in which they circulate? Nonetheless, as we probed this kind of thing, we found out that this kind of shift is not unique to loneliness. In fact, the more people started looking at other kinds of psychological or social or cultural or economic or even political contexts, the more apparent it became that a similar kind of shift takes place just about any time a human being feels threatened or even merely uncertain for an extended period of time. Once you feel precarious for more than a few, moments to hours, it turns out you don't have to ask for this. The body automatically turns on this essentially molecular defense program and starts subtly reshaping how the immune system is going to do its job by reshaping the transcriptome of these immune cells. So in the ensuing decade or so, where we've been kind of chasing down this phenomena, we now understand quite well how this comes about. This particular profile is mediated predominantly by fight or flight stress signaling, activation of the sympathetic nervous system in response to experiences of threat or uncertainty, which ends up being represented at a biochemical level as the release of neuroeffector molecules such as norepinephrine at sympathetic nerve terminals. That norepinephrine has an opportunity to change what a white blood cell is doing by interacting with beta adrenergic receptors. This particular shift that I've described here seems to be mediated predominantly by beta-2 adrenergic receptors, which are hooked up to an evolutionarily sculpted wiring diagram that essentially calls on transcription factors to increase the transcription of genes involved in inflammation like IL-6, IL-1, TNF, while at the same time reciprocally inhibiting the transcription of genes involved in type 1 interferon responses. These effects are mediated predominantly in the monocyte pool, to a lesser extent in dendritic cells and neutrophils. And in addition to changing the molecular stripes of the existing monocyte pool, these dynamics also actually change the population structure of the white blood cell pool itself. And that happens as these contextually triggered experiences of threat end up activating those sympathetic nerve fibers. And one of the places those sympathetic nerve fibers release these neuroeffector molecules is in the bone marrow microenvironment, which is busily regenerating the white blood cell pool every day. As you guys probably know, large numbers of myeloid lineage cells, monocytes, neutrophils, probably dendritic cells as well, are lost every single day. And so our bone marrows have to continually replenish those lost myeloid lineage cells. That replenishment process is, of course, carried out by hematopoietic stem cells. And when norepinephrine is released into the niche that these hematopoietic stem cells occupy, that essentially reshapes the production of the hematopoietic growth factors that govern which kinds of progeny these stem cells make. So, for example, as the sympathetic nervous system is more active, you get essentially effectively a cytokine or a growth factor mediated vector towards making more myeloid lineage cells at the expense of making more lymphoid lineage cells. So less T cells and B cells come out, more monocytes, neutrophils, dendritic cells. And as a consequence, you now have a bone marrow piling up this sort of different set of cells, and those end up then entering systematic circulation throughout the body and essentially create a circulating white blood cell pool that is a little bit thicker with monocytes. Those monocytes tend to be less mature, more pro-inflammatory, less effective in mounting antiviral responses. If these monocytes get sort of recruited into damaged neural tissue, they tend to produce bigger inflammatory responses. And so neurodegeneration is more likely to take place. They produce bigger atherosclerotic plaques in your coronary artery because they're more effective. You can think of them as like wound healing or scab production cells. They tend to produce hyperinflammatory responses in the airway, so you're more likely to get sort of serious COVID or pneumonia or asthma under these circumstances. These monocytes in particular are key antigen presenting cells, so they also essentially provide advice to the lymphoid lineage cells about what kind of a response they should make. Should they make a response that's effective against extracellular pathogens, a Th2 or 17 style response, or should they be making a response that's more effective against intracellular pathogens, like a Th1 response? These monocytes in general will pull for a Th2 or Th17 response, and so essentially undermine a response that would be effective against a virus or a malignancy. And then, of course, these monocytes also play a key role in structuring how the body responds to an initiated tumor. Once you've got genomic damage driving the sort of growth of a primary tumor, as those cells squash their surrounding healthy neighboring cells, monocytes come in responding to that tissue damage. They don't find any foreign microbe responsible for this, and so they execute a wound healing program that inadvertently drives metastasis, producing lots of blood vessels, growth factors, basically remodeling extracellular matrix, taking the molecular seatbelt off of these cells so they can spread throughout the body more easily through these newly recruited or built blood vessels. So essentially what's happening here is our body seems to have evolved a sort of a programming in the immune system that says, when I'm feeling threatened or uncertain, take my default bias towards fighting viral infections, which is an important default virus for social creatures like ourselves, take that and lighten up on that and instead clear out some molecular resources to more effectively battle extracellular pathogens, particularly the kinds of bacterial infections that would have historically been associated with experiences of this sort of fight or flight stress or acute stress. So this whole program, of course, is makes perfect sense if the things in our ecosystem that are correlated with sympathetic nervous system activity look like this. In other words, if the things that cause us to feel fight or flight stress responses are also things that are likely to wound us, then biasing the immune system towards more effective wound healing responses makes perfect sense. In fact, you can imagine how this would easily be incentivized, right? If you get an immune system that can, to some extent, anticipate when we're going to get wounded, it will, all things equal, do a better job of preserving a body than an immune system that sits around waiting for a wound and then starts to try ramping up, let's say, an antibacterial response. Of course, the problem with this programming is that we don't deal with a lot of that stuff these days. What we do deal with is this chronic, low-grade drizzle of threat and insecurity. And that's driven not by things that are going to physically wound us. It's driven more by things like this. In other words, these you can think of them as closer to like cognitive stressors or symbolic stressors, things that are not going to bite us or gore us. They're not going to, sort of exfiltrate our gut bacteria into our sterile tissues. Nonetheless, our acquired programming is still pumping out these fight or flight stress signals. It'll be at low levels, but chronically over time in a kind of a time context that's very different than we were actually originally evolved to deal with. And so as a consequence of that, you get over decades more molecular, you can think of it as like fertilizer for the production of metastatic cancers, neurodegenerative diseases, and atherosclerotic plaques. So on some level, because of this mismatch between the ecosystem we evolved in ancestrally, in which this programming makes some reasonable sense, and the world that we live in now, where we've ingeniously engineered all kinds of ways to feel threatened and insecure constantly because that's so economically productive, this becomes, in some sense, a recipe for how not to live, both figuratively in the sense that these risky environments are unpleasant, but also literally in the sense that if we run this molecular defense program, we're basically leaving the iron on all the time. Irons are great for smoothing out wrinkles, but if you leave them on all the time, they're going to burn down the house. And that's what's happening, is this chronic activation of the system, not necessarily at maximal power, but at, you can think of it as unprecedented temporal scope over time. That seems to be kind of a recipe for increased disease of the sorts I just described earlier, increased mortality, that kind of thing. So as we were thinking about this, as the kind of the first maybe 5 to 10 years of the work had come in, and we had a much better sense of what this molecular programming looks like, we could look at anybody we chose to and see, do they look like somebody who's feeling threatened or insecure, regardless of what they actually say or what we told them, and can we block these things with adrenergic antagonists and so on. So we started to get a pretty concrete sense of how this system works. But from the kind of the basic science or the preclinical work, we do those things with like genetic tricks to, make a new mouse that lacks this, or pharmacologic tricks to, deafen this particular receptor system. These don't really seem like viable solutions to this question of how we should live instead. And so we started looking in the context of these risk factor studies to find what you could think of them as bright spots of resilience. We were very interested in finding people who had low levels of this molecular threat signature going on in their immune system, despite the fact that they were in ecosystems where, in general, most people have activation of these systems, and then trying to understand what's special about them. What are they doing differently that actually allows them to run a healthier, RNA profile in their white blood cells, despite being in these threatening ecosystems? So, of course, this question of how we should live our lives has not historically been the provenance of molecular biologists. This is more typically the preoccupation of, you can think of them as moral philosophers, or maybe they're modern day equivalents of happiness psychologists. So I actually happen to know a couple of happiness psychologists and was asking them, how do you guys think about well-being and resilience and the factors that protect a body from a dangerous ecosystem? And as they were, sort of answering this, I was basically in their minds, I was asking them, what role does happiness or well-being play in opposing these things? And they said, well, we're happy to help you with this. We've got all kinds of good measures of these things. You just have to tell us what kind of happiness you're interested in. And I was kind of flummoxed by this because I'm a grant-funded biomedical researcher. I have no direct acquaintance with happiness in general. The idea that there's two kinds of happiness was even more of a, sort of puzzle to me. So as we started thinking about how to answer this question, they basically mapped out, when we think about well-being, we distinguish, and this is not something we do recently, this is something that, Aristotle did when he was thinking about the nature of the good life. So this distinction between, you can think of it as what they call hedonic well-being. That's the type of happiness that comes from feeding your own neural reward circuitry, eating tasty food, seeing beautiful sights, watching a funny movie. These are all examples of relatively direct activation of, you can think of it as a mirth or a happiness circuitry in the brain. And we actually understand these circuits neurally pretty well. So that has, though, historically been described. You can think of this as like the Epicurean recipe for the good life. Just keep eating tasty food and having fun. And, you sum up those happy moments, and that's what constitutes a good life. The alternative perspective that stretches, again, way back, at least to Aristotle, probably much farther back than that, is this notion that there's a different kind of happiness that comes from pursuing some kind of virtuous goal for the world that transcends your own immediate self-gratification. So this notion that Aristotle called eudaimonic well-being, finding the truth in you, the person you were meant to be, the world you were meant to create. So this idea that striving after a better world, even if that is just taking care of your family or running a business or discovering something in nature, communicating some deep truth in art, these kinds of, sort of, you can think of them as stretch mode, creative or generative versions of happiness are experienced somewhat differently by people, but they also, we know, have different neurobiological substrates in the brain and therefore potentially different impacts on what the body is doing. And we'll come back to this later, but just to give you a sense of how the happiness psychologists are thinking about these things, they'll ask people questions like, how often do you feel happy or satisfied in your life, to assess hedonic well-being, whereas assessing eudaimonic well-being more has the flavor of like, how often do you feel like your life has a sense of direction and meaning to it, or you're growing and becoming a better person. So this more kind of indirect, I would call it almost like flowery or, more, I don't know, mental or virtue-driven version of happiness. So I'm gonna kind of cut to the chase in the interest of time and just kind of point out that when we did studies measuring these two types of happiness, as you might imagine, they're pretty well correlated, not perfectly. You can find people that are, sort of have high levels of hedonic well-being, but relatively low levels of eudaimonic well-being. They got happy by essentially consummatory self-gratification. And then there's people that are relatively happy and they got there apparently largely through eudaimonic means. They're saying, yeah, I'm doing something important for the world. I'm growing, becoming a better person, creating a better world, that kind of thing. When we look at people who have relatively high levels of eudaimonic well-being, we see actually exactly what Aristotle might have predicted, and certainly, probably your grandmother would have predicted. When you do good in life, your body, is pretty happy about that. So we're seeing relatively low levels of inflammatory gene expression and relatively high levels of type 1 interferon gene expression in those individuals. Interestingly, when we look at people who have high levels of hedonic well-being, but relatively low levels of eudaimonic well-being, they're feeling good, but they got there through a different path, they actually look pretty poor. They have relatively high expression of inflammatory molecules, relatively low expression of type 1 interferons. And this is true even if we control for or take into account and move off the table some of the consummatory self-gratification pathways that you might imagine would impact molecular physiology, like eating a lot of food or smoking or drinking. So even when we account for those things, there still seems to be this residual sense that this life of what one collaborator of mine called cheap, empty calories happiness is having some kind of different trickle down into the body. Now, this is originally sort of a both fascinating and provocative, but also almost, like too charming and picturesque result. So, as you might imagine, we kind of chased this around for a while, making sure, is this just like some weird one-off thing that we happen to bump into? And in fact, this seems to be a pretty robust phenomena. So you can see these kinds of dynamics repeatedly across, both demographic, strata in our domestic research studies. We can see it in, people living in different cultural conditions, Japanese, businessmen working in an IT company, kids in Colorado hanging out in video game parlors all day long. Let's see who else. Korean seniors in a sort of a nationwide study of sort of mid and late life well-being. So this seems to be a reasonably reliable correlation. But that said, it is a correlation. And of course, we mistrust correlations. There's all kinds of correlation and causation issues. So as you might imagine, I spend a lot of time these days running around trying to figure out how we can experimentally induce more eudaimonia and see if we can get some kind of causal consequence in terms of changes in these threat-related gene expression signatures. So I'll show you just like a quick rundown of a couple of examples of this. In the left panel here, you can see the change in a sort of like a one-number summary of that threat-related gene expression signature in a group of older, predominantly African-American, predominantly grandmas living in South Central LA who have started in on an intergenerational mentoring program called Generation Exchange, where they get out of their house, stop watching Judge Judy, and go into the local school district and start serving as teacher's aides. They get trained during the summer and then allocated to a classroom. And it's, a reasonable amount of engagement. It's several hours a day, so it's not a trivial lift. But nonetheless, over time, they tend to show a pretty reliable decrease in this threat-related molecular signature. And we especially are impressed by this because this is actually the largest effect size we have for the antiviral component. So you can imagine in the beginning of the COVID epidemic, we were particularly interested in, is there something distinctive about this kind of classroom mentoring, either symbolically or even just in sort of microbial epidemiologic exposure dynamics. It's actually helping them have such, sort of favorable profiles. And again, also note that this is like about maybe a 7 to 8% reduction. So we're not talking pixie dust here that will make cancer go away or, sort of magically protect you against all threats in the world. They're just the things that trim an immune system for more effective function and produce effect sizes that are about the size that we see for social risk factors in, you can think of it as social epidemiology more broadly. So on the panel on the right, you see results tracking the change in the same molecular signature for people who were living in the Riverside area around UC Riverside, where another collaborating psychologist was sort of running a community-based study in which she randomized people to do essentially random acts of kindness for either the world in general or for themselves or for some specific other living, breathing, real human being. Like, don't just save the whales by sending in money or write a poem or something like that. Like, find a person and actually help them. So you can see the magnitude of change over time here compared to people in a control group who are just recording their daily activities and not doing any acts of kindness at all. So you can see control group, no significant change over time. Random acts of kindness for the world, also no change over time. Random acts of kindness for yourself, sort of that same trend towards more adverse profiles, higher threat signature in people essentially doing, kind of the hedonic well-being lifestyle. The people who are looking better are the ones who are doing random acts of kindness for some other specific living, breathing human being. And as they replicated this result, they did more intensive measurement, and it turns out that a lot of you can think of it as the experiential correlate of that favorable change, seems to come from some combination of self-reward for doing good for others and actual overt social appreciation of somebody's contributions. So these are examples of the kinds of things that, are these like protocols we can just distribute to the world and we'll automatically make disease go away. No, these are, sort of the first steps. You can think of them as like behavioral engineering tests to make sure that these things are really causal effects and not just correlates of some bundle of virtue that also, sort of affects biology in other ways. So I'll share with you another somewhat provocative result that came up for reasons that will be kind of obvious in a moment. Particularly came to people's attention during the COVID epidemic. So we had for years been doing studies of loneliness and social isolation, which are to some extent different. You can be lonely in a room full of people. You're not isolated, but you may not know those people or trust them. And so you still feel disconnected from them. And it's the loneliness that actually drives the toxicity, because togetherness, the antithesis of loneliness, is essentially a chief safety signal for the human brain. So a brain that doesn't feel like it's in a context with other people that care about them and will, help them be well. Even if you have no demand at the moment, just if you're absent that, these systems will come on automatically. So we do monkey studies where we, sort of, again, try and experimentally shape an animal's social context and see how that changes what's going on in their circulating white blood cells. And of course, we love monkeys because we can get to their bone marrow and their lymph nodes and all kinds of other places where we be able to kind of fill in the story about where does the change in the circulating white blood cell pool come from. So we've been doing a study like that where we took, sort of adult male rhesus macaques out of the field cages that they live in, which typically have around 80 other monkeys, kind of all hanging out in their monkey tree houses and rooftops and stuff like that. And we put them in their own little monkey apartment, basically. So this is actually an ample apartment by monkey standards. Usually they put the monkey in one cage, but just to make sure that there's no unreasonable stress here, they gave the monkeys a second cage to play in as well, give them toys, stuff to do. It's not a bad life. If you look at stress hormones in these animals, they're not particularly perturbed in a, sort of like a major way, but they just don't have other monkeys around that would fulfill this social safety signaling. And so even though we're not depriving the monkeys of food or, mobility or, entertainment or anything like that, when we look at blood parameters in these animals, we'll see exactly what I was describing earlier. They bump up the production of those classical monocytes, these kind of young, immature, pro-inflammatory monocytes, and they reciprocally bump down the level of type 1 interferon gene expression in their circulating white blood cell pool. So the interesting opportunity we had here, though, was to look at what kinds of interventions we might use to buffer this. So these, inadvertently turned out to be, sort of like a model of shelter in place. This idea of like, okay, you're plucked out of your normal social life, you're in your house, and, you're just hanging out there, basically. So in this scenario, you can see the changes in these immune molecular profiles persisted for at least a couple of weeks. After the kind of 2-week window in which they were tracking the physiology, they took the monkeys out of these little shelter-in-place shelters and returned them to their field cage, whereupon they reverted back to their baseline state. And then a month or two later, they took the same monkeys and locked them down again. Only this time, they were locked down in the same kind of cage, but in the cage next door, there was a juvenile monkey, probably about, analogous to maybe a 4 to 6-year-old in human terms. And when you look at the molecular responses the animals had during this second lockdown that came with a companion, you see a considerably different profile. So the level of classical monocytes in circulating blood actually trends down, not up. And there's no net loss of this kind of type 1 interferon molecular signature. Now, this is not the perfect study because, having a juvenile monkey there as sort of like a playmate or a caregiving object is confounded with the number of times that you've been locked down. So there's always limitations to these kinds of things. But clearly, something about this is changing the way these monkeys' immune systems are recalibrating in this shelter-in-place period. And I'll share with you for a moment the reason we think this might actually be due to caregiving circumstances. And that is related to what we've learned about the neural pathways through which eudaimonia operates. So there seems to be at least 2 you can think of them as physiologically distinct, but probably conceptually related biological mechanisms going on here. One involves changes in the way the central nervous system is operating during periods of, you can think of them as prosocial engagement or eudaimonic activity. So remember I told you that these molecular signatures are shaped predominantly by activity in the sympathetic nervous system, beta adrenergic signaling, all that kind of thing. So we, so far have not found any eudaimonia magical hormone or something like that that competes with threat neurobiology at the level of the, sort of the target cell at the end of the stage, at, sort of in the immune system level. What we seem to see is more the activity of some system in the central nervous system that is basically blunting outflow through this stress response system. So the way we understand this kind of values-seeking or values-driven neurobiology is largely shaped through analyses of a circuit in the brain that's generally referred to as the ventral striatum. It's kind of this dopamine-mediated system that especially motivates or animates what you would colloquially call seeking, hoping, wanting behavior. So this system, which is comprised of things like the caudate, putamen nucleus accumbens in particular, this dopamine-rich circuit is basically the seat of, sort of scanning your environment, finding something good, going after it, organizing behavior for that. You can think of it as a prospectively shaped reward system. It rewards you for doing good stuff. And it's to some extent distinct from another reward system in the brain that was kind of more classically sketched that rewards you for essentially consuming good stuff. In other words, there's a surprising degree of neurocircuitry distinction between hedonic and eudaimonic well-being. The interesting thing about the ventral striatum, this kind of seeking, hoping, wanting circuitry, is that it is known to exert lateral inhibition over the threat-related circuits that actually drive sympathetic nervous system activity out of the brainstem. So this capacity essentially for an active value-seeking circuit in the brain to sort of laterally veto stress responses that are coming out of this threat detection and response system in another wing appears to be the reason that we're getting less of this threat-related molecular signature. In other words, there's within experience or within the brain, there is like this tug of war between, am I going to feel threatened and frightened and insecure, in which case we get that signature, or am I doing what I should be doing what I care about, doing what's most important? And if so, don't be threatened. Just go on, do what matters. So part of the reason we believe this system is, I would say involved, we don't think it's probably the only thing, but there's a striking correlation between how strongly this ventral striatum circuit activates in response to goals that are subjectively meaningful and what's going on in the circulating white blood cell pool. And this is an unusually high correlation in behavioral science terms, but nonetheless, it seems to be kind of recurrently observable. So the particular scatter plot I'm showing you there comes from a study where a very bright neuroscientist came up with a paradigm where she would basically expose people in an MRI scanner to sort of visual stimuli that evoked either things that they cared about, had already identified as subjectively meaningful, basically goals or aspirations, and compared how strongly the ventral striatum reacts to those things with the magnitude of ventral striatal response to subjectively sort of significant goals. It's not like they're bad things. It's like not my particular thing that I care about most. So the more your brain in this seeking, hoping, wanting circuit activates in response to something that you care about, the lower your circulating, sort of, you can think of it as like your basal pro-inflammatory gene expression set point is. So that's part of what makes us think that this is important. And of course, again, we can't directly manipulate these things at the neuroscience level, at least in human beings, but you can, bet we run around all the time, with chemogenetics or something like that, messing with the brains of mice to try and make sure that this is really what's going on. If we just activate that brain circuit is enough to block this threat-related molecular signature, it is. If we sort of underactivate or inhibit the activity of that system, does that allow the threat system to have, more free reign over circulating immune cell gene regulation. Yes, it does. So that's one thing that seems to play a role. There's a second layer of this, and it may actually be somewhat functionally connected. We're still not totally sure about this. But remember, as I was talking about the monkey study, and to some extent, also the South Central LA grandma study, the kind of the intergenerational mentoring, and even the random acts of kindness study, they all, to some extent, involve what sort of neurobiologists of behavior would call caregiving. There are different ways of deploying your resources on behalf of others, either these school kids or the random act of kindness target that you've been, sort of like you selected to, sort of do your assigned work for the day, or, the charming little monkey kid next door that you're gonna, play with and take care of while you're, otherwise sitting through this sort of rattling lockdown. So these are all examples of this sort of caregiving concept that appears to have significant neurobiological underpinnings. Most mammals, basically any animal that involves an extended childhood and needs parental maintenance. In other words, you don't just deliver your kid and then he instinctually goes out there in the world and starts eating and living and stuff like that. If you have to take care of them as they grow, that doesn't come for free physiologically. So organisms have developed a fairly elaborate set of adaptations. Some of them are obvious, like lactation. Some of them are less obvious, but especially structuring the behavioral components of this. One area that people talk about a lot is the parasympathetic division of the autonomic nervous system, which is opposed to the sort of stress threat driving sympathetic response. The parasympathetic response, by contrast, is kind of a rest, digest, and increasingly, theorists believe, rest, digest, and give care system. That in fact, it is a tending and a befriending system. It's a way of allowing you to take essentially your physiological resources and lend them to the well-being of another. This obviously, ancestrally involved for progeny, but the argument is that you can take that same neurobiology and generalize it to taking care of your spouse or your neighbor or your community or your nation or this cause or purpose. In other words, the same neurobiology can be hooked up to much broader and more abstract concepts. So this caregiving system, like I said, is running, at least in part, its physiological impact is mediated through activation of the parasympathetic division of the autonomic nervous system, and that is well known in physiology. We all kind of learned this in kind of neuroscience 101, that antagonizes sympathetic nervous system activity at the peripheral level, and not just at the central nervous system level in reducing the amount of central drive to threat signaling, but also peripherally in terms of physiologically antagonizing the kind of molecular push generated by the sympathetic division of the autonomic nervous system. And that is why when we look at indices of sympathetic nervous system activity, things like respiratory sinus arrhythmia, the extent to which the vagus nerve sort of changes heart rate as a consequence of changes in breathing, that is a parasympathetically mediated connection. And so people in research neuroscience often use that as an index of just how strongly at the tonic level this inhibitory system works. So individual differences in respiratory sinus arrhythmia in terms of a sort of a molecular readout of parasympathetic nervous system activity are reasonably well correlated with essentially better, particularly antiviral response. We do see from study to study sort of some variability in the magnitude of the effects on inflammatory gene expression, but the type 1 interferon system in particular seems to be very sensitive to this kind of system. So those two mechanisms end up being, sort of an explanation for how these differences in the way we live might potentially end up impacting what's going on at the molecular level. And what I'm going to wrap up with today is sort of a relatively recent evolution in how we're trying to deploy this particular concept. So far, I've been talking mostly about protocols that we're administering to groups of individuals to see how their molecular physiology changes on average. But for any given individual, some of these things will work better than others. So for example, we know that you can drug these systems. We know that you can deploy other means to, let's say, reduce sympathetic activity or increase parasympathetic nervous system activity, things like wellness practices, meditation, Tai Chi, yoga, that kind of stuff. And we also know that there are these sort of eudaimonic routes to reducing these things. For any given individual, one or the other of these may be more powerful. If you ask me to meditate, it's not going to do any good because meditation aggravates me. I can't sit in one place doing nothing all the time. It's like not going to work. Other people bliss out. They just think it's the greatest thing on the face of the earth. So I'm going to have a different molecular response to, a meditation protocol than might my, friend Mike, who, built his entire research career around documenting the molecular virtues of meditation. So with an eye towards helping people actually realize these kinds of things in their own lives and helping select the changes that are most, favorably impactful for them, we've started asking questions not so much about how humans in general should live as questions about how specific individual human beings should live. And the way we do that is by collecting data on the conceptual approach, the dream scenario. I'll describe one. You will see that we're not totally to the dreamland yet, but the dream scenario is that we're monitoring what's going on in people's bodies more or less all the time, and bouncing that information out of the body and into some environment where we can make sense of what's going on at the molecular level. Obviously, that will be helpful for medicine, particularly detecting, sort of the prodromal syndromes that precipitate disease before a diagnosable disease actually onsets. You can imagine how this would be great for all kinds of other analyses related to epidemiology, policy decisions, how are people doing in general. But what we're especially interested about is this notion of using this information and giving it back to individuals to help them actually shape their lives in the way that is most favorable for their molecular well-being. So you can think of this almost as like genomic biofeedback or molecular self-awareness or something where we can take this molecular information that for science purposes we can get to. It's not impossible to get this information. It's just like so far, we haven't had any reason to present it to people if it doesn't have, an immediate health implication. But partly as this work becomes more kind of socialized through, sort of articles about it or books that it gets involved with or whatever, eventually we get, almost always when someone publishes a book that has an angle on this in it, we get people coming and saying, hey, can you do that for me? I've got this terrible situation. I know it has something to do with my life. I can feel that. I just don't know how it's coming about and what I can do, for that kind of thing. So it was actually this sort of, sporadic clinical need, really more than anything else, that led us to start working up this infrastructure. So what we do in these sort of molecular self-awareness studies is we will take repeated blood samples from individuals over time. This is a modestly ambitious one, but we've done more than 100 over a course of a year or something like that. We will quantify that level of molecular well-being, which is just the reciprocal of the profile I was talking about earlier. This is basically what molecular well-being is: low inflammatory gene expression and high type 1 interferon gene expression in kind of a relatively ratio metric. So you can see that individual kind of bouncing around over time. What we ask people to do in the context of these studies, in addition to taking blood samples, is measure characteristics of the life that they're leading. So this was a particularly ambitious young woman who was measuring all of these different parameters in her life: when she was sick, how good this day was, how many steps she took, how much money she spent, what was the temperature of the weather outside, how much time did she spend outside. You can see tons and tons of metrics there. And I would expect most of these are not terribly predictive. And so what we do is we take this stream of numbers that she's generating, quantifying essentially her life circumstances, and we feed it to, essentially machine learning algorithms to pull out the specific components that are most predictive of her waxing and waning molecular well-being. In other words, we don't care about any other individual. We want a lot of repeated measurements from this person to know what in her life circumstances varies favorably with her molecular well-being. So in her particular case, these are the parameters that actually ended up mattering. Was she eating food that she felt was healthy? Was she spending time outside? Did she get a lot of exercise? Was she socializing? And how much time did she spend at work? She had a particularly bad work situation, and that turned out to be very interesting, as she actually eventually explained it to me. this work situation involved being a commissioned salesperson, a newly commissioned salesperson. So she's sitting here competing with pros over a limited piece of the pie. It's exactly the kind of insecure, if not outright threatening ecosystem that so recurrently comes up as a predictor of kind of general adversity. In addition to mapping that aggregate well-being thing, we can also kind of break that down into its two constituent components of inflammatory gene expression and type 1 interferon gene expression and start to pick apart what matters for this molecular tranche in this individual versus that one. And interestingly, it's not the case that the same things matter. What matters for inflammation, in her case, for instance, often involved like sleep, general well-being, mindfulness, kind of reduced stress, and not eating fat, basically. What mattered for type 1 interferon biology was a somewhat larger list, some of which overlap with what mattered for inflammation, but also some parameters of which are relatively distinct to that molecular system. So we're pretty sure that there's more complexity to the way these systems are regulated based on experience than we've captured so far with this simple concept of threat. And this is how we're going to kind of chase that down and try and figure out which of these tranches of abstract information flow into the molecular system are, how are they being transmitted at the physical, sort of cellular and molecular level. So what we're basically, the next step, so this so far has just been essentially taking information out of the body and figuring out how it works. But the obvious next step is, making changes based on that to see whether we'll get consequent changes in molecular well-being as a follow-up to that. So that's more or less the end of my talk. I used to end my talks by, just making the point that, like, in principle, the way we experience life doesn't just stay in life, it gets all the way down into the nucleus of our own lives, into the molecular composition of our bodies, and to the extent that it changes RNA profiles that produce proteins that have an average half-life about 2 to 3 months, that's kind of the tail that we're working with in terms of experience changing physiology. If we kick off a positive feedback cycle that, institutes a new regime in your body, which happens sometimes, we can see that, maybe the effects will be even longer than that. So we used to facetiously say, hey, don't mess up, go live your life the way you're supposed to. But now, we're starting to be in this point where, like, it's not so facetious. We could actually make changes or at least be aware of how we're leading our lives in ways that affect these things. So in some senses, we're thinking about, am I leading the good life? Am I leading the life I was meant to lead? Asking our molecular physiology whether it thinks we're leading the good life. It's like a very bizarre idea on some level, but on another level, I mean, if you think of the human genome as having helped us survive and thrive for millions of years when we had no cognitive philosophy to work with at all, maybe it has learned a thing or two about how best for us to be in this life. And so with that, let me thank you for your attention. Thank the people that have supported this work, and I guess answer any questions or complaints that people have.
Dr. Cole, thank you. That is absolutely amazing. Essentially, it sounds like you're providing, we do lots of wellness surveys around here, and it's all subjective, right? I feel that, and that's very meaningful and important. Right. But it feels like you're almost creating an objective way, and not almost, you are creating an objective way to assess that.
Yeah, although I have to be careful because there is important well-being that is just experiential. I mean, in my view, molecules exist mostly to support, I don't know what you would call it, mental or spiritual or experienced well-being. But there's clearly a shadow of that world. And if you can keep the infrastructure, the physiological infrastructure for that well-being healthier longer, then yeah, you're absolutely right. Both the objective or I think of it is objective, but it's most importantly, it's kind of like physiological or infrastructional. So it's kind of like a additional thing that you'd want to get a more comprehensive, holistic view of well-being. Exactly.
I'm getting, so thank you for the questions that are popping up on Zoom. If there's any questions in here, Argavan, Dr. Salas, I'll turn over to you first in person, then I'll move online as well.
Thank you so much. This is really fascinating. And you talked a lot about two different types of kind of essentially positive experiences, eudaimonic and hedonic experiences. I'm curious what you've studied or are aware of related to negative experiences. So if we snap at somebody and then feel bad about it. Or we witness someone snapping at somebody and it's upsetting to us. I would imagine that probably heightens these like inflammatory markers, but I was just curious what you've seen on that.
Yeah, great question. So the closest thing to a simple answer we have to that is if it leaves you feeling threatened or insecure for an extended period of time, then it will get you there. And it's actually an important distinction because, you know, if you snap at somebody and they're not of great consequence for you, it doesn't threaten your interest that much. It's not your best day ever, right? But at the same time, it's not, you know, sort of anywhere near as impactful as if you know, snap at, you know, your love at a fragile time and it changes the course of your, you know, personal relationship history, right? So it really is more about two things. One, how much this threatens your core interests, and two, the kind of the temporal scope. So the other thing we know is like a bad day does almost nothing to these systems. The human neurobiology was built to eat bad days. It can even eat bad weeks and months. We can get through bereavement without a wholesale collapse of our physiology. Many times, that's not always the case, but to a first approximation, it is lifestyles of threat and insecurity that are the big drivers of these sociological or, you know, social epidemiological impacts. And so we really have, you know, come to not worry about individual snaps or individual insults or things like that, as long as they don't fundamentally change your view of yourself or the world or how trustworthy other people are, that kind of thing. So having that kind of a, you can think of it almost like as a meme about what's going on, because these are all concepts we can't directly measure, but as a kind of heuristic for shaping how you might do future research to kind of probe around that theory. This idea that extended threat or uncertainty is the key thing that you can hook almost any experience in life up to. So it really depends on, does the snap threaten long-term, you know, safety, essentially.
I have a question here from Rebecca Merrill. Rebecca is a trained psychologist, a personal coach, runs one of our huge called Making Space personal development coaching leadership course. She's saying, you know, this is amazing research. She was texting me throughout. This is unbelievable. She has a question about, you know, through your experiences through the studies and whatnot, you have any comments? I would love to know how to inspire lonely, isolated people to do more other focused caretaking, especially if they're depressed.
Yeah, this we answer this question over and over. So obviously, you know, I don't have the magic solution to this, but one thing we've learned is that it's almost impossible to sort of, you know, psychotherapeutically attack loneliness head on. And that's because loneliness, you know, acute loneliness that comes from moving to a new city or whatever generally is kind of self-healing. The kind of loneliness that's really toxic is chronic loneliness, which comes from a sense of mistrust of other human beings. And lots of times those are not accidental events. They're based on, you know, real life betrayals. Trying to talk lonely people into trusting other people, it's hard. They've got good reasons for why they're doing, either related to betrayals they've experienced or related to just personal sensitivity, like when small things affect them a lot. And so they're just kind of filtering out risk by keeping other people at arm's length. So it's really hard to attack loneliness that way. But there's a great oblique route to tackling loneliness, which involves, instead of treating people as, you know, sort of like pathologically afflicted individuals, it basically involves going to them and saying, what do you care about? You know, get them to start thinking about stuff other than themselves, because self-focus is tremendously, you know, toxic and kind of downward spiraling. And instead get them to think about what they care about. And interestingly, a lot of lonely people end up lonely because they believe that they care about things or experience things differently than other people do. They feel like no one knows the world, no one sees the world that I see, no one experiences this kind of thing. And so if you can get people sort of to identify the things that are personally important to them and then to start working on them, one of the funny things is it's almost impossible to achieve anything really significant by yourself. So if you can get people who are starting off kind of atomized and mistrustful into even just a very small community of other individuals who care about the same thing, that is magic. It is totally amazing, because you start to take this very feeble, exhausted, seeking, hoping, wanting system that those folks have, and you give it a little spark of life. Yeah, maybe there are some people that care about what I care about. And, you know, that's often good at kind of gradually building trust. And as you build trust around shared interest in saving the whales or whatever, that kind of trust can bleed to, like, if I needed a cup of sugar, who should I ask? Maybe one of these fellow whale savers or something like that. So I realize that's not a specific program, but this overall abstract strategy of, like, back off from trying to make the bad stuff go away, try and find the good stuff, start reigniting that, and then let the natural social synergy that comes from focusing on aspirational, seeking, hoping, wanting things, kind of do that work. It becomes an energy that helps you re-socialize, basically, and build trust.
Wonderful. We have a lot of questions. Sorry, we have a lot of questions on Zoom. Dr. Grimes asking, chronic stress eventually induce epigenetic change that permanently alter gene expression?
I am not a big believer in permanence of anything. So I think epigenetics in general has been misinterpreted as implying more permanence than is really there. There are definitely permanent things, like a germline mutation. That's a relatively permanent thing for a lot of people, right? I mean, that's a thing that will shape whole bodies and, to some extent, destinies. But we have been particularly impressed by plasticity as the sort of the rule rather than the exception. And this is to some extent based on studies where we've taken fairly traumatized populations, sometimes correlationally in humans, more often in experimental monkey studies, and reintroduced them into more stable, supportive environments and quantified, how many, like literally in this white blood cell pool, how many genes wandered outside the normal range of variation in their activity levels in the monkeys that were, taken from their mothers at birth and put in a cage with a bunch of other infant monkeys, which sounds charming, but is really essentially the Lord of the Flies. So it's just like chaos and instability and dominant stuff everywhere. Very hard on the little infant monkey system. So monkeys raised that way for a few months, the equivalent of maybe 3 to 5 years in human terms, they end up with many hundreds of genes that are operating differently, at least in their white blood cell pool, almost certainly stuff in their brains, but we don't have the brain tissues to actually make sense of that. Then you take those monkeys and you just return them to their normal social colonies. There's just some stability, some parental oversight, and more, I believe it's around 95% of the genes that were kind of systematically deviated revert back into the normal range. There's a few that are kind of locked out, but the recovery is more the rule than the exception. But what's interesting about those monkeys is they still show more variance over time. They seem to be twitchy. So they seem to have developed at least some molecular neuroscience that just leads them to be more reactive to environmental circumstances. So it's definitely a mix. It's not like one is true and the other one's not. But the plasticity opportunities, I think, what I'm concerned about is people feeling like, hey, my grandmother had some catastrophe, I'm cursed. So, the epigenetics is going to like leave me locked out. And we just don't see any evidence of that. So
we have a lot of great questions. Do you mind if we go a few minutes over?
Not at all.
Wonderful.
Absolutely.
Dr. Valentine asks, given the evolutionary context of innate immune system, do you anticipate or see significant variability across ancestry groups in the molecular wellness markers you described?
That is a great question. So in principle, I agree. In practice, we haven't found too much so far, but neither do we have studies of the scope that would allow us to, I think, accurately address that. So I believe in principle that some of these, essentially kind of ancestral / genetically transmitted variations in reactivity should exist. And we have done some studies with, for instance, SNPs in the promoters of cytokine genes that clearly do affect how connected that gene's activity is to environmental threat. So now that I think about it, there's at least one case where that has been shown to be true, and that is something that is the particular SNP we were looking at is relatively restricted to Northern European ancestry individuals. So probably yes.
Echoing this is incredible, supports Buddhist principle of altruism as a source of happiness or contentment. Is there a difference between the virtual other focused activities versus in-person caring? Yeah. So online friend groups are just interacting in person.
Yeah, this was, as you imagine, a big topic during pandemic periods. And I was lucky enough to have collaborators who really loved the online world. Even before the pandemic, they were, they just loved this idea that people were living out these symbolic experiences and these amazing symbolic experiences that transcend what's possible even in the real world, in the online world, and that might somehow, sort of at least, measure up, if not exceed, the impact of real world socialization. But alas, we did not find any evidence for that. So online prestige, online sense of community, we found relatively modest associations with these molecular well-being markers. And we continued even during pandemic periods to find that people who, were relatively more real world integrated showed more favorable molecular profiles. So it's hard. And this is something I think we learned during the pandemic. It's harder to replace real world presence than any of us would have imagined. And on some level, that I think has been a huge missed research opportunity to really plumb what is it about in person that is so different that it ends up producing different physiology.
I hope you can study that in the future. That's really interesting. Great question by Dr. Golden. That this one might be applicable to a lot of topics these days. How will AI be involved in the nuclear activity or affected by the list of multiple things? Might AI play a big role in really a lot of this work? And
yeah, certainly as a research infrastructure, it definitely will. There's a lot of, you can think of it as knowledge borrowing we need to do. Right now, when we do these individual studies, we're treating each person as if they were the first person ever born and just trying to induce from their own thing. But we would love to be able to do something like template matching, where we had large assemblies of people and we could almost sort of like cluster map. Oh, I can feel even now after only 20 observations that this person's regime, gets them near this cluster versus that. So there will be a huge amount of kind of research infrastructure stuff for that. Then there's a separate question about how AI's social impact dimensions will affect human well-being. And there's, as you guys know, a lot of doom scrolling around AI, replacing human beings and stuff like that. But honestly, AI is crappy at the stuff that human beings are good at, and vice versa. So I'm characteristically optimistic stance for me. I actually am not at all worried about that kind of stuff, just because, steam engines didn't, reduce the need for human beings. In fact, it probably left them healthier because they don't break themselves as often trying to lift heavy stuff. So
I might have one more in here just because it's coming from, I believe, Dr. Scaff. He's just asking, have you studied physician burnout with this data?
We have. So there's a great line of work on that comes from Carter Labaris, I think is her name, at UCSF. And she is a surgeon who studies surgical trainee burnout, which is, surgeons are notorious for being like these high octane, live fast, die young, drink heavy, these kind of high risk, high reward temperaments. And she's got, very clear evidence that interventions can help with that kind of, she's another one of these like mindfulness lovers. So she's got this whole mindfulness-based stress reduction protocol for surgeons, specifically tapped into their sort of the glorious idiosyncrasies that leave them, more vulnerable to this kind of stuff in the first place.
Well, Dr. Cole, I have endless comments in the chat, in the Q&A, text I got directly saying, this is amazing. And I sometimes get it, but this is next level number of comments about this. This really is wonderful. I do hope you can come back again in the future and present more. This is something we're definitely interested in.
I'm always happy to do that. Absolutely. Yeah, this is, I was here 7 years ago. It's even better now. So good work, you guys.
Maybe less than 7 years from now when you come back.
Sure.
Well, thank you for those of you who came in person, as always, really appreciate it. And thanks for the big showing online, as always. Thank you, Dr. Cole, for joining us today.
Pleasure.
Have a great rest of the day.
Yeah.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.